Welcome, everyone. Thank you for attending this discussion of MIT's response to the pandemic. My name is Ian Waits, and I'm the Vice Chancellor for Undergraduate and Graduate Education, former Dean of Engineering, and an MIT faculty member in Aero and Astro for 30 years. And for the past 2.5 years, I've played a role on several teams that have helped MIT to manage the pandemic. And my goal here is just to set up the topic, and then I will hand it off to our moderator and panelists to actually do all the hard work. But I wanted to start with just a couple vignettes for you. The first is a memory I have of walking down the Infinite Corridor in mid-March of 2020 after we've made the decision to send our students home, and it was largely empty. And students were out on Killian Court with borrowed robes taking photographs that would become their ad hoc graduation photos. And so it was just a very disruptive, painful sort of moment to sort of evacuate the campus. The second image I want to paint for you is the Johnson Ice Rink set up as a field hospital for our potential use and Cambridge's potential use. It was never used in that way. And now let me contrast that with our laboratories establishing 24-hour-a-day schedules so that they could observe density restrictions but never stop their important work. So MIT never stopped its research and research educational enterprises during the pandemic. The fourth vignette is to have you think about 2,000 teaching staff over a period of 10 days taking 1,250 classes and moving them online. And these are, yes, lecture classes, but lab classes, project-based classes, people sourcing and mailing out kits of hardware to people all over the campus. So it was every bit of a heroic activity on campus. And in the last 2.5 years, we've learned a lot. We've learned how to communicate differently, both locally with Zoom, but also to the MIT community, again with Zoom. We have 8 a.m. calls that will share information with as many as 500 people. We've done 150 of those in the last 2.5 years. We've also learned, though, the value of doing things in person instead of on Zoom. So it's been wonderful in the last year or year and a half to get back to many, many more in-person activities. We learned the value of having top-notch medical and emergency management teams, but perhaps even more so, we learned the value of having a top-notch IS&T team at MIT. Just about everything you do these days, if you're directing the actions of 30,000 people, has to be delivered on one of these. And so we have an exceptional IS&T team that stood up all of the enterprises that enabled us to manage the campus for the last 2.5 years. And over that time period, we've done 1.6 million PCR tests for our community, which has enabled us to keep our enterprise going fully in person. We learned the value of deep scientific inquiry with the rapid advance of mRNA technology for vaccines, much of which drew on fundamental work that was done at MIT in the years prior to the pandemic. And one of the things that I learned about pandemics is that the hardest science is not biology, it's behavioral science. At least from the perspective of managing a community of 30,000 people. The biology was often easier to predict than the behavior. And so it was heroic, but it wasn't special. We did this, many other people were undergoing the exact same things that we did. But I think we tackled it in a very MIT kind of way. We remained nimble, we made mistakes, we fixed them. We shared all that we could with the community. We borrowed when we could. We invented when we had to. And we kept our really important teaching and research enterprises going at essentially full steam for most of the last 2.5 years. So you're going to hear from the panelists about some of the impacts of the pandemic, but the most important things for MIT will be what we carry forward into the future from it, both the things that are positive and the things that are very negative. So before I hand it over to them, though, I want to say a few words about the panel's moderator, Professor Pekka Hosoi, who's down on the far end. So like many of us, Pekka was not appointed to a formal role in helping to manage the pandemic. At least I don't think so.
Not my fault.
And I think she's not someone prior to the pandemic who would have regarded herself as an expert in public health monitoring.
Correct.
or management or modeling. But she emerged as someone who I and others relied upon very heavily. And that's because Pekka has a very special talent. Even within an MIT community of very, very bright people, Pekka knows how to build parsimonious, reduced order models of the physical world. And what I mean by that are models that have just enough parameters in them to capture the important first-order behaviors, but not so many and not so much complexity that it obfuscates the important trade-offs. Right? And what's important about that is that Pekka can not only describe those models to me, a fellow engineer, but I can turn around and describe them to others, whether it's MIT leadership or community-based calls and things like that. So Pekka became our go-to person for modeling trade-offs on testing policies, masking policies, filtration of rooms like this, many, many more things because of this special talent she has for building parsimonious reduced order models. To say it very specifically, she was one of our secret weapons in the pandemic, so thank you, Pekka. So before I turn things over to Pekka, though, I do want to thank all of you for being here, for supporting this marvelous institution, and for being part of our shared pursuit of making a better world. Pekka?
Thanks, Ian. Thank you for that lovely introduction. I have to say, the reduced order models that I developed, the talent that Ian is talking about is actually counting. I can turn it into a counting problem, which was, I think, like Ian said, it made it very clear what kinds of decisions we should be making. But let me introduce our fabulous panel today. Today we're going to be hearing from Mei Hong, professor in chemistry, Chris Love, who is a professor in chemical engineering and a member of both the Broad and the Ragon Institute, and Professor Eric Klopfer, who is a professor in the Department of Comparative Media Studies and also the director of the Scheller Teaching Education Program. And everybody has done, I think I'm really excited about this panel because everybody has done something to impact our understanding of the pandemic or to move through the pandemic or to make things easier or to prepare for the next pandemic in different ways. So before we get to that, I just want you to know who these folks are. And so I'm going to ask everybody just to give just 1 to 2 minute introduction on who you are and what your research looked like before the pandemic, just so we can see what the transformation looked like. So I'm going to start with Chris, and I'm just going to go down the line. So Chris, take it away.
Thanks, Pekka. We have this idea here of convergence here at MIT of like biology and engineering as they come together. It's been coined by some of our colleagues here, and I guess I'm kind of that person. I trained as a chemist and immunologist. My lab sits in chemical engineering. We operate out of the Koch Institute on cancer research, and we develop technologies that are important in human health. Both to characterize how diseases and interventions are manifest in patients, but also the technologies necessary to bring those interventions to reality through manufacturing. So we've had the opportunity to work on technologies to analyze cells at the single cell level. We built some of the first technologies in that space, some of which in their later iterations were actually important during the pandemic to discover some of the antibodies that were used. And we've been very interested in the last few years in small footprint manufacturing for medicines to make them more globally accessible.
Mm-hmm. Terrific, terrific. And actually, I'm gonna do one follow-up question on that, because as a mechanical engineer, I'm guessing that the word manufacturing evokes a very different image in my brain than in your brain, because I think of cars. Right? So give us a little bit of a picture of what you mean when you say manufacturing.
Yeah, so from a chemical engineer's perspective of manufacturing, I think this means thinking about how do we actually make the molecules at scale and bring them to a stage and purity that's appropriate, in our case for medicines, could be for other purposes. We're particularly interested in making biologic proteins and things like insulin and monoclonal antibodies, vaccine components. So this starts with some living system that produces that component, plus a bunch of other things it naturally makes, and then figuring out how to whittle out everything that's not the thing you want, and making sure that that's in the vial at the end of the day.
It's perfect, perfect, great. May, tell us about your work.
Yeah. So I'm a biophysical chemist. And I'm trained in physical chemistry, particularly the method of NMR spectroscopy, which is nuclear magnetic resonance spectroscopy. It's the molecular underpinning of MRI that you come across in hospitals, magnetic resonance imaging. I use these large superconducting magnets to study biological molecules, particularly proteins involved in infectious diseases. My lab also is interested in studying proteins involved in neurodegenerative diseases like Alzheimer's disease. So anything which is not well soluble, so membrane-bound proteins in viruses. And so before the pandemic, we studied influenza virus, an important membrane protein for many years, really tried to understand the structure, molecular structure, as well as the mechanism of action of that protein called M2, which is involved in how this flu virus would infect host cells. And so that line of work prepared us very well for the pandemic, because we just basically pivoted to study the corresponding ion channel in SARS-CoV-2. So that's my lab's work. The NMR spectroscopy part is my physical chemistry background, but I always like to apply it to proteins in important for human diseases. So yeah.
Great, thanks, May. And Eric.
Thanks. So I'm coincidentally a biologist by training. My work appropriate for today many years ago was on ecology and evolution, but I did a lot of mathematical modeling of biological systems, including epidemics. That's a long time ago, and since then, for the last 25 years or so, I've been doing work in education and educational technology. I run our teacher education program here, which trains MIT undergrads who want to be math and science teachers, and run a lab called the Education Arcade, which focuses on using sort of progressive pedagogies and technologies to promote STEM education in the pre-college audience. We do a lot of stuff on games, simulations, project-based learning, computer science education, and project-based learning sort of overlaps a lot of that work. Our research methodology is design-based research. So in design-based research, we start with real problems that people are facing, whether it be problems in learning that teachers are facing, or management that they're facing, or particular conceptual difficulties that students have. We start with those problems and we think about ways that we can use technology to help solve those problems. And that ties into a lot of the educational technology that we develop here and draw a lot upon diverse students across the Institute.
Great. And by the way, this is the first time I'm learning that we have a lab on games on campus, so I'm definitely going to be visiting that. So now that we sort of set the stage, let's all think back to, again, March 2020, where everything changed, March 2020. And like Ian said, I'm now thinking about with the visuals I have from March 2020, and one of the things that sticks with me is that everybody wanted to help in the MIT community. Like, you came in and students, postdocs, faculty, everybody was trying to figure out ways to take their technical expertise and use it to make some progress in this giant challenge that we were all facing. And like Ian said, that the labs were up and running, and I mean, honestly, we couldn't keep people out. Everybody wanted to be there working to make some headway. And I think we have some exemplars of this here. And so I'm now going to ask you guys to think about, so if you go back there, all of you pivoted your research in some way when the pandemic hit. And so what I'd like to hear about is how did your research pivot? What is the work you did around COVID? And maybe even give people a sense of what it was like to work on campus, or what are the unusual challenges that we were facing during COVID. And I'll go now in reverse order. So Eric, we'll start with you, and maybe you can talk a little bit about sort of the tools that you developed to help parents and students during this time.
Great, thanks. Yeah, so as you said, like everybody else on campus.
Sorry, I have to interrupt.
Yeah.
Sorry, I'm already screwing up my duties as host. So two things. One is at the end of this, we are going to open up for questions, and this is how you submit a question. So on your phone, if you go to this, we'll put this back up in the background in a minute. But we're also going to move to the next slide, which is going to show a little bit about some of the work that Eric's been doing. But I'll put the question slide back up when Eric's done talking.
Thanks. Thanks. So like literally everybody else on campus, I went home, and some of my colleagues went home to think about what could we do to help, and we knew that like people were stuck at home, kids were stuck at home, parents were stuck at home, teachers were stuck at home, and with a lot of variable resources at their disposal. Some of them great. In my case, in fact, you can see that's our logo that we had there. And I'll give credit to my two kids, Oren and Ari, who developed that for our website. They were in high school and middle school at the time. So they had me at home. I was able to sort of help enrich their learning and help them engage in gaming and things like that effectively. But there were lots of students, teachers, and parents across the country who were not. And so I worked with a lot of folks in open learning and MyLab and really across MIT to help develop a set of resources, which we ultimately called Full Steam Ahead, which was a set of online resources for everything from pre-K through grade 12. It was materials that parents could use with their kids. It was materials that teachers could use with their classes, either sort of in a focused way or sort of give to them as enrichment activities. It was materials that they could use either sitting at a computer and do things online, or they could give things where in a lot of cases where it was materials that teachers could send home, or parents could do in their houses with materials they might have around the house. And we ultimately developed 10 modules, really drawing upon so many different parts of MIT. We did stuff in space exploration, artificial intelligence, and the very first unit was on COVID and epidemics themselves. So really trying to give people a handle on what was going on. I mean, as Ian recalled and you recalled, we were also confused at the time, and we were trying to sort of help educate people at the same time we were trying to help them understand the circumstances around them. And ultimately, I can talk about it later, but this became a set of resources that's been really durable, and we've reused both sort of for local programs where we were running after-school programs from campus, our virtual campus. We all worked from our homes, as well as programs that we've run around the world.
Very good. But, one quick follow-up. How do people get engaged with that? Like if there are teachers who want to use your resources, how do they interact with you?
Yeah, so we set up the website. The people could just sort of download the materials, use the materials freely online. We created at the time we had a bulletin board that we could sort of post things in and share things and share ideas with people. They could contact us directly. And that's how we wound up working with really programs around the world. So people found these materials and they said, can you help us customize them? Can you help us use them with our set of teachers in this country or this community? And they found us and we were able to respond, I'll say with a lot of support from the Institute.
Yeah, that's great. I have many follow-up questions, but I'm going to move on to May. May, tell us about your pandemic experience.
Yeah. So in February 2020, my group published the structure of the M2 protein, this proton channel that's important for the influenza B virus, B virus. Now, every season, flu season, in the fall, you get typically influenza A, which is the one responsible for all the historical pandemics. But in the springs, the influenza B infection surges. So we decided to work out the structure, the atomic resolution structure of that influenza B M2 protein. It's a proton channel. It took us about 1.5 years, one of my best students in my career, to really figure out how to clone the protein, how to express it, how to purify it to enough yield that we can actually do NMR experiments. And then, go to the 900 megahertz, 800 megahertz machine, measure all these spectra, figure out the structure. Right after that came out, the pandemic started, and we were like, at first I was in denial mode. I thought, man, this coronavirus can't really be that much worse than flu. But when that became obviously wrong, I told my flu team, we've got to pivot. So we took what we learned about solving the influenza B M2 structure, all of that expertise, moved it right over to study the corresponding ion channel in the SARS-CoV-2 virus. And SARS-CoV-2 has 3 membrane proteins, only 3. And some other structural biologists got to the spike protein structures really fast. The spike protein is the one responsible for getting that virus into the cell. But once inside the cell, the virus has to make its own genetic material, its own protein, and so on. And then this E protein, this envelope protein, is responsible for budding that new virus and get it out of the cell to infect another one. And that E protein, envelope protein, also conducts calcium. And that calcium conduction activity is what makes this virus cause all sorts of inflammation risk, problem to the host cell. So if you can block that E protein, you've got an antiviral drug potentially. And so we need to know the structure in order for medicinal chemists and pharmaceutical scientists to actually design small molecules that work better than the current small molecule, which don't bind with particularly good affinity. And that's the goal of our experiment. And so we solved that structure in about 6 months. It was a time shrink from the first, the influenza BM2 work by about a factor of 3 because we were just prepared, basically. So very hard work during the pandemic because, I can only have one person coming in. It has to be a postdoc, not a grad student. So the grad student was, zooming in with the postdoc, trying to figure out how to make this protein, all the advices along the way. And I was kind of, I was told, okay, I can only have so many hours. And, I was just coming in anyways. And Plasma Science Fusion Center is where my lab is situated. And thank goodness they don't think about NMR very much. They think about nuclear fusion. But our superconducting magnets are all there. And so they knew how important it is to get those virus proteins structures solved. And so they were very supportive. And I needed to be there with my coworkers to just debug along the way. And we got it. We were lucky. We got it to work. Yeah, so that was very exciting. Hopefully, now we're collaborating with more people elsewhere to really medicinal chemists now to try to move on to the small molecule drug discovery.
Yeah, really. Well, it's amazing because I've been in so many. Ian can attest to this. I've been in so many COVID meetings with so many people. This is the first time I've heard that it's the E protein that's responsible for inflammation, which is really interesting.
Yeah.
So
there may be other players, but this one, it's known that the calcium imbalance in the cell is. This protein is doing the ion conduction. Yeah.
That's super interesting. So presumably that has implications for long COVID then as well.
That's right.
Yeah.
That's right.
Yeah. Interesting. Chris.
Yeah, so we had been working with the Gates Foundation for several years prior to the pandemic to think about reducing the cost of vaccines globally. So today, vaccines are all manufactured, coming back to manufacturing in very large facilities. There's a couple of them in the world that make the majority of the vaccines that are given pre-pandemic globally to kids everywhere. And every year, the WHO sets targets for what those vaccinations should look like for all these other diseases besides COVID. And every year those targets are missed, and it's because manufacturing fails in a lot somewhere. So we've been thinking with them about how do we use kind of these small footprint manufacturing concepts we've been building to break the economies of scale by scale out instead of up. And so we built some of these automated systems that we could put yeast in, a bit like brewing beer. You put yeast into it, and it fully automates from the time you put it in to having formulated protein basically ready for injection on the other side. It's a few other things to put it in a vial, but the materials necessary to do this. And we developed this with DARPA and the Gates Foundation. And then, everybody says March. For me, it was February. So we got Happy Valentine's Day from the Gates Foundation. Can you help us build a vaccine candidate for low middle income countries that would be appropriate, using some of the technologies and ideas that you've been building? We said, sure, we can help. So they emailed us the sequence, and everybody likes to talk about the mRNA. Super fast, really amazing technology developed here. I'm just going to say that the 4 guys that I had in the lab afterwards doing this work, in 29 days, they went from having the sequence to having yeast strains ready to go for manufacturing, a draft process, and materials for animal studies. So, smaller resources, but it was amazing how fast they were able to go. They talked about, Ian's walking down the infinite corridor by himself. We're in the lab. There were the only 4 of them, the only ones on the whole floor in the KI. And they said it was like completely weird just to be there by themselves. And we all kind of have an understanding now of what like interactions can be like and how we can be safe about those. We didn't know. So they were trying to figure out like how could they be in the bench space around this and doing the work that they had to do. And I think just very interesting. And then everybody in the lab that was at home trying to help order and coordinate things so that they could get the things that they needed to do to keep pressing forward with these candidates. Super exciting. We did the work and it was developed. We were able to transfer it to 2 developing country vaccine manufacturers that both made GMP lots with these materials. We went forward in a multi-institutional collaboration with one of them to early stage clinical trials, and the company that's now taking that forward is the largest vaccine manufacturer in the world, and trying to figure out how to position that for next generation vaccine candidates here.
Yeah, that's incredible. And so this also reminds me, as May and Chris are talking about what it was like to be in the labs during the pandemic, like you said, in the early stages, we didn't know. Like, think back, we didn't know. Could you put 2 people in the lab at once? Do we need to be wiping down all the surfaces? Are masks okay? There was a lot of stuff that was unknown at the time. And, but people were still coming in to figure it out. And I do remember, and Ian probably remembers this too, that you had to figure out who were the essential people on campus so that we could give people like Chris and Eric and May space in the lab. So remove all of the unessential people so that they have access to those facilities. And what I learned is that there are 2 essential groups of people on campus. There are the people that feed the animals and the people that feed the students. And the rest of us are expendable. So yeah, and then gradually we learned more and we figured out what were the right practices in the labs. I do want to come back to something that was really striking, I think, in all of these answers, which is how fast we were able to move. Right? I think typically when you think about the academic experience, it's a very it's a long-term, precise kind of, nobody thinks of us as speedy in general. And, but these things were turned around just at record pace. And I think May touched a little bit on why that was, but I want to emphasize that. So maybe I'll start with you on this one. Why is it that we could do all of this so fast?
Yeah. So I think the first thing is the students and postdocs. They want to work on this. I mean, I don't need to tell them second time, oh, let's find the ion channel in SARS-CoV-2 and work that out. They want it. They jump to it as fast as I can, and they don't want to take a break. They didn't want to take a break. Because my building was largely empty, just like Chris, and these NMR rooms are big because these superconducting magnets take a lot of space, and so actually the density is not an issue, but it's kind of lonely to have only one person wandering down the hallway setting up the experiments. And so they were just going all the time. And it took us 3 months to get that protein cloned and purified, and then you don't know whether it's going to spectral quality, whether it's actually amenable for structure determination. And we got lucky that this protein E behaved very homogeneously structurally, and so the spectral quality was excellent. But without the expertise from the influenza M2 research over the decade, right? We wouldn't have been ready. I mean, how do you make a membrane protein? How do you express it? It's hard. It's not like a water-soluble protein. These are very hydrophobic proteins. Bacteria don't like to express this, thing. You have to add another protein which is water-soluble. What other protein do you add to make the whole thing? And then purify that whole thing and then cut that extra piece so that you can end up with the hydrophobic virus membrane protein of interest. All of that is, knowledge and expertise came from working on influenza when it was sort of a regular project. I mean, people know influenza is important. On the other hand, there are a lot of other things that are important. So thank goodness NIH was supportive over the years and kept in mind that there are pandemics looming around the corner. And so now my group is working on a bacterial transporter. And after you talk to people for a while, they might forget or get a little blasé about, oh yeah, antibiotic, resistance, but it is a problem, So I just hope that, there's enough public-private support for fundamental research about antibiotic-resistant proteins involved in that, about virus proteins. Now, coronavirus is big for my group. We're just putting most of our thinking on the coronavirus front. There are going to be other coronaviruses we're going to look at, not just the SARS-CoV-2. Yeah.
Chris, you also, I think, had a similar experience that this was of why you were able to move so quickly.
Yeah, I mean, I'd kind of just reemphasize some of it. I mean, I think it was having a goal, it was being prepared, and then, I think a little bit of luck. And, on the goal side, right, obviously, when we had our mission, we needed to make a vaccine, go after that, along with, many others who were thinking about very similar questions. And that gave the students something to focus on, to kind of put to practice the things that we had been thinking about. The preparation, broadly in the field, we were very lucky in that this is a virus that we had prior knowledge of in other related coronaviruses that gave an insight into how to think about the vaccine work. It had already been developed in concept in other MERS, SARS-CoV-1. So having that as a place to start from and build. From our perspective as a lab, we had been working on these small footprint manufacturing systems for about 7 years at that point. We had prototype systems that we had built. I mean, I kind of lovingly call them chewing gum and duct tape prototypes because they're kind of those MIT things you kind of expect. We bought a bunch of stuff off the shelf and then ripped all its brains out to make it actually do what we wanted it to do. We had started a consortium with the industry in 2018 to start adapting the yeast organism we use for production to have it capable of making certain kinds of proteins that are important in vaccines and biopharmaceuticals. That turned out to be important because in a pandemic, you don't have time to build a new manufacturing facility. So for us to transfer strains to someone to use it, this particular yeast organism requires typically methanol for its carbon source. And as a chemical engineer, you kind of look at it, oh, it's no big deal, it's just methanol. It's explosive when you put it at very large volumes. And not every facility that's designed to make vaccines has the capacity to handle that safely. But through the work we'd been doing in this industry consortia that we had started, we actually had built some of the biology and tools to adapt that host to 8 other carbon sources using CRISPR-Cas9 and some of these other cool synthetic biology tools to enable it to be able to do that. So we could transfer those strains out, and they could take our strains that we had built in the lab, and they went to 1,200 liters and made material for clinical studies in like 2 weeks. It was amazing to see. And I love, we have this nice mass spec of our stuff out of the lab, and it has all these little peaks on it. And then there's the one from the 1,200 liter reactor. It's the same little peaks.
Nice.
Yeah, we got that one right. Yeah. So it's kind of the combination of all these things. And I think, platforms like the mRNA technologies for manufacturing, these were things that existed prior to the pandemic. And if they had not, we would not have been able to respond as quickly.
Right. Right. Yeah. No. This is something that really strikes me in hearing all of these stories. It's that in some way we were lucky, but in some ways we were prepared. Right. That all of this, that we were able to get strains in 29 days or structures in 6 months because there had been years or decades already invested in that. Right. And if we had not done that work on flu, if Chris had not done those work on small factories, we couldn't have turned this around in the time that we did. So even though we didn't know exactly what was coming, we were working in areas that were close enough that were adaptable, that you could adapt on a short time scale, which I think was really, really critical. So let's see. I'm going to switch gears a tiny bit, so I think this is some of the I would say the triumphant stories that we have out of the pandemic. The pandemic also exposed a number of weaknesses and inequities that we have in the system, which now that we're aware of, we should probably think about what those are and how to plan moving forward. So, Eric, let me start with you. Talk a little bit about your work and about, I think in particular, inequities that were revealed in the studies that you did.
Yeah, let me just make a quick comment on the last one. I'll echo everybody's sort of sentiment around people being so important. And the preparedness, I think, that we had in our space was a community of people who were already sort of working on problems in K-12 education, as well as our work in online education that we'd been doing, so we were ready to do that. And we had administrators throughout MIT who wanted to support this work. So that was really great. In terms of the inequities, I mean, the things that people sort of immediately sort of jump to, which were really apparent right away, was the sort of technological inequities that we had. Everybody was sent home. Everybody was learning on Zoom or Google Meet, and what happened was some kids didn't have broadband. Some kids didn't have Chromebooks. Some kids didn't have cameras that worked, and when that was your means of communicating, that made a huge difference in those kids' lives immediately. The sort of less apparent stuff, and there's a couple different groups. One in my group that studied elementary education. Emma Anderson led that one, and Justin Reich's group created a paper called Imagining September, along with a colleague at Harvard, looking at sort of K-12 education more broadly. Where were these gaps and what had happened? And the biggest gaps were these sort of social gaps that happened. That kids lost their connections with other kids.
Yeah.
Kids and teachers lost their connections. And in some cases, you were able to reestablish those or sort of help those. So in cases where a teacher had a small number of students and they had good technology, they could check in with the students. They could sort of see how they were doing. Maybe there was a parent at home. In other cases, you had kids who had parents that were worried about how they were going to put food on the table the next day because their jobs were gone. They had poor technology. They had teachers who were teaching 200 students. Those students lost those social connections. And it was just tremendous. The other thing is, and we were all talking about this beforehand, is kids who are in sort of critical stages. We talked about it. We see it in our freshmen now, and other colleagues all across the country. The kids who sort of like had an online junior and senior year of high school are not ready for college in the same way that the other students are. And all sorts of other transitions. You were talking about grad students who also sort of missed out on critical periods. So those inequities we see in different kinds of cohorts, and there's a question of how they're going to make that up. Are they going to be sort of left behind as people sort of before them and after them wind up sort of filling in those gaps, or are we going to find ways of sort of helping them catch up? And it's not just sort of catch up academically; it's catch up in terms of time management, in terms of critical stages of being an adult.
Yeah.
And learning to have those connections. So it's a problem that I think we still need to work more on.
Yeah, I totally. I see something really similar in my classes actually. The juniors, so the juniors who were just away for 2 years, we teach them the technical stuff, which we always teach them, and it turns out there's a whole bunch of other stuff that they learn in college just by being in dorms and being around one another, which they now don't know. And so it comes out in strange things like when they're working in teams in classes, and they literally don't know each other. So it's I 100% agree with that. That's something that it's going to propagate through the system, and we just have to figure out how to handle that. Chris, thinking about weaknesses or inequities that we've seen in the system that the pandemic revealed.
Yeah, I mean, I think on a more global scale, at least from the perspective we've worked in vaccines, the obvious one is just distribution of those vaccines to everyone. And I think while we've had the benefit of having access to a lot of them here, there's still a lot of parts of the world that this has not been the case, and I think that's a matter of both price and availability and just sort of the nature of some of the vaccines. I mean, the cold chain and distribution of these things is a real problem, and how do we build better solutions that are more equitable there? I know we were talking a little bit about this before, but I think one of the interesting things is if you take a map, and probably everybody's seen one of these maps of the distribution of vaccines globally, there's regions of the world that are underserved in that way. If you take a map of manufacturing capacity for medicines like vaccines, that map looks almost identical to it. So maybe we'll come back to this later, but I think it says something about what we need to think about in terms of where manufacturing solutions are available and being able to produce goods, not just for medicines, but perhaps other intermediates and food parts, materials.
Yeah.
So maybe something we can talk about in a little bit.
Yeah. Perfect. I'm gonna remind everybody to submit your questions because I'm gonna turn it over to you in a minute. But before we do that, I'm going to actually, Chris, I'm gonna let you follow up on that thought, because the last question I'm gonna ask you guys is what's next, and I'm gonna ask this in a very broad sense, so you can talk about whatever you think, whatever you'd like to say, looking forward into the future on whatever time scale. So, Chris, I'm going to start with you so you can finish that thought that you just started.
Well, actually, so I'll broaden it even just a step further, and I'll kind of bring it back to our experience a little bit in developing these vaccine candidates. In that, we as a globe have become very good at interacting among different parts of the economy in different ways, but it also creates this reliance on the logistics and supply chains of those and our manufacturing systems to provide everything, food, parts, plastics. If you had owned a Sartorius plastic bag that's used in vaccine manufacturing during the pandemic, and you had one under your couch somewhere, you could have gotten a very pretty penny for that on eBay. Just saying, plastics and the raw materials that we need to do these things were actually in quite short supply and still are today, actually. So if we think about the dependence that we've created in this kind of global economy, but then in a pandemic that starts to break down because things like air transport doesn't happen. So we made those strains in 29 days. It took another 29 days for us to find a flight to get them out of the country to the vaccine manufacturer. What if those strains and the manufacturing capacity for being able to do that had been on site regionally to be able to do that? We could have just sent them the instructions for how to do it, and they could have recreated it there. And so I think, as we look ahead, the thing that I see is like where we need to continue to focus on is, centralized manufacturing has certain value in economies of scale, but our automation and intensification and things that we're learning across actually all disciplines about how to be better at manufacturing and doing it in an efficient, small-scale way is something that, as we think about what platforms we might need next, we're going to need this, and it's not just for medicines, but probably for all materials.
Yeah, and I mean, it's really interesting because in so many ways, but we've been hearing so much about virtual and about computing, and all of this has been the center of our attention for a long time. But the fact is we live in a world of things, right? So, these things have to come from somewhere, and there are a lot of challenges that are just logistical challenges associated with that. Yeah, really interesting. May, looking forward, what do you see?
So looking forward, for my area of research, in order to continue to figure out protein structures involved in disease, the technology has to keep up. And the spectroscopy that I do, this NMR, which is the molecular underpinning of MRI, that takes these large superconducting magnets, and it's a big shared equipment, and that's the area which now gets squeezed a bit for the whole field, not just MIT. Because resources are limited, there are other structural methods, but other structural methods often have a hard time getting to these very small virus proteins. And when I say other structural methods, I'm referring to X-ray crystallography or cryo-electron microscopy. All of these involve large equipment, and so I try to tell the federal funding agencies and anybody who will listen, right? Spectroscopy, molecular spectroscopy like NMR should be supported. The technology, took decades for the whole field to develop, and my lab is one of these that develops technologies. But without the equipment, it's kind of hard to, do very well. To solve this structure, you need large magnetic fields. That gives you the sensitivity and resolution. And in Europe, where research is supported more because they're putting the resource into this kind of work. While in the U.S., we're seeing less of that. So you can just tell the science, without certain cutting-edge equipment, it's hard to address good biomedical questions. And that's where I see, I hope, the government will continue to support, but private sector, also would be wonderful. So I'm sitting right at the intersection between technology and biomedical, I see that's really needs to happen for us to do our work.
Yeah, and I think that comes back to our discussion about being prepared, right? These are not things that you can suddenly buy when a crisis happens, right? The fact that you had not only the equipment, but you had the personnel that were trained to do this, that you had postdocs and graduate students who knew how to do this was critical. Eric.
Yeah, I'll pick up on that point of preparedness and reflect on a little pre-pandemic work we used to do. So for many years, we did these classroom simulations of epidemics. We sort of simulated in the classroom, and people would sort of learn about things like latency and probability of transmission and incubation and all these sorts of things that now everybody sort of has some intuition for based on the pandemic. But at the beginning, they really didn't. And so particularly some of those things actually were very problematic. How could I give this to somebody else when I wasn't symptomatic? And so I think some of those simulations were really effective in getting those ideas across. And so the question is, what are the experiences we can be giving them today now to prepare them for the next thing? And we think about things like climate change and working with artificial intelligence and misinformation. These are sort of other problems that are here today and sort of impacting the way we work. And so it's about sort of giving them experiences with those to sort of help them prepare them. I think the other thing as I think about is social learning online. I mean, we've all been on a lot of Zooms, and it was certainly it was the best thing that we had available to us, but it really doesn't facilitate social learning, and that's been such an aspect that a lot of people have missed online, and I think it's something that we can add more of. And I don't know if it's us all being in the metaverse with headsets on, but I think there's other ways to think about ways that we can facilitate social interactions and social learning online.
Yeah, terrific. I have a bunch of really great questions from the audience. I know they're going to cut me off, so I won't have time to get through all of them, but I'm going to try to get one for each of you. So let me start with this one. And this one, I think maybe we can start with Chris. What are the advantages of using yeast to produce vaccines versus other platforms?
Yeah, it's speed and cost.
Okay.
So I had a chance to spend my sabbatical at Biogen several years ago and looking at what would transform manufacturing in terms of low-cost production and the speed with which we can develop it. And the answer turns out to be the biology. And it's for reasons that it has impact in places that you don't necessarily anticipate. The biology itself is often the thing that makes the proteins that we're trying to use for production, but that impacts everything else, all of the quality assays, everything else. So just one example, yeast doesn't harbor viruses the same way that mammalian cells do. I don't have to then test for that.
Yeah.
And there's allowances now and regulatory practices for that. So yeah, I think it's this combination of the biology features, but then the impact that it has on the rest of the manufacturing strategies.
Nice. Let me. I'm sorting. There's questions coming in, which is great. Okay, Eric, I'm going to give you the next one here. Communication turns out to have been one of the big problems of the pandemic, and convincing people to get vaccinated. In Full Steam Ahead, do you have lessons about how to reach out, and do you have suggestions for how scientists and policymakers can communicate better with the public about science?
That's a really good question. I mean, so part of certainly the epidemiology unit of Full Steam Ahead was about that, specifically about the epidemic. We were early on, and so it was really about preparing people to sort of understand information as it came in, but it was all drawing. If the platform consisted of, for each of the modules, we talked to experts at MIT. So it was not. Yeah. It was we did activities, we had hands-on activities, but it was a lot about bringing that information from experts in these different fields that we could use to communicate with the world through this. And so we had experts in epidemiology, but we also had experts, talking about space exploration and other things. I think it's something that we can and should continue to do, and I think about it with two hats on. In fact, one is my hat in education that I have, where I primarily do my work. I'm also head of the Department of Comparative Media Studies and Writing, where our science writing program exists, which is all about communication to the general public about these issues. And I think it sits in a really important place as what I will say the premier institution for STEM work in the world. It's about sort of getting our messages out in ways that are understandable, and it's not always about having the best science, but the best way to communicate that. And sometimes that's through experience, like the stuff that we develop in games. Sometimes it's about analogy, metaphor, research. There's lots of different ways we think about that communication.
Yeah, just actually a quick follow, because you mentioned that you had this activity about people simulating epidemics or understanding as you turn it up. When people go through that exercise, does it change their attitudes about vaccines, or is that something you measure?
So we didn't have vaccination in that one, but it's certainly changed their behaviors in terms of keeping some distance if I don't know what's going on. So if I don't know if you're healthy or sick, I might keep some distance from you. Even you could see it visually. The simulation, just so you know, it's actually people walking around in a classroom interacting with each other. So you really see them doing this, and that understanding is very it's very visceral. So you see someone who gets sick, and you're like, oh no! And it also gets into the ethics though. Like, do you want to shun that person, or do you want to think about how you help that person, or so there's a lot of great discussion that goes on around that as well.
Yeah, and we had a lot of discussions about that in MIT policies, right? So how do you keep the campus safe, but you don't want to single out people in a way that they can't participate in the community? So how do you handle those trade-offs and make policies that incentivize the best behaviors? Yeah. May, I've got one for you. Do you know whether the E protein has evolved as quickly in the new strains as the spike, or is it something that is conserved across the different variants of the coronavirus?
It has not evolved very much, actually, so that makes it a good target. So the Omicron, one of the earlier Omicron variants for the E protein's transmembrane domain, one of the residue threonine is turned into an isoleucine. So we're thinking about that will be, maybe studying this variant sequence and see how the structure gets changed. So E protein, like the influenza M2 proton channel, these ion channels are evolved much less quickly compared to the fusion protein, viral fusion protein. So the spike is a fusion protein that gets changed and mutated easily, and so the vaccines become, don't work very well when it changes. Just like you hear all the flu strains, H1N1, H5N1, that's all hemagglutinin doing the changing. The M2 is pretty conserved. And that makes these ion channels good antiviral drug targets. Yeah, they're not gonna prevent you from getting sick, but if you get sick, and you hopefully will take a pill and block these proteins, yeah.
Wow, that's really interesting. Let's see. Eric, I'm gonna give you one more, and then I'm gonna read one for everybody. Okay. So Eric, what are ways that local schools, say in Cambridge, can quickly implement the social learning improvements that you suggest?
Oh boy. So I think I'll say two things. One is through full steam ahead, and with the support of it was the chancellor at the time, who's now our provost, as well as the Office of Experiential Learning, supported outreach into the local communities. So we ran summer programs, we ran after-school programs. So a lot of it was about our direct outreach to those students and teachers. I think the answer is that it's not a quick change. I think there are some fundamental challenges that we face in schools today, and maybe the pandemic has been a chance to sort of reset and look at those things. What is learning? What is durable in learning? We talk a lot about assessments that are sort of superficial and trivial. Students who remember particular math facts or definitions of words, which are important things, but they're not going to sort of prepare people for the future of learning and the future of work and the future of sort of civic participation. And so I think we're in the process of sort of going through a slow transformation, which is facing backlash in some places about what does learning look like in the 21st century, and we're well into the 21st century now. And it's got to be about thinking about problems like the next epidemic, climate change. How do we get kids to really think about these challenges and how they're going to face them in the future, and not just focusing on some of the real basics?
I heard a great quote actually from one of my colleagues on that, which this is your contrasting knowing facts versus knowing how to think. And she said, we're teaching as if there was no Google,
Yeah,
right?
yes, yeah.
So I think that's a transformation that we're
absolutely.
making and have to continue. Okay, I'm giving a gold star to the person who sent in this question. This is my favorite question, and it's a hard one. And each of you are gonna get a shot at it. So, okay, so here's the question. If you could immediately change MIT policies and culture, what are the most important things you would do to institutionalize your learnings about solving problems more quickly and effectively? You have 10 seconds before I call on someone.
Is this being recorded?
I'll go in this order. So Eric, you're gonna go first.
All right. I'll be a little daring here. I think as we, I talked about transformation in public schools in the 21st century. I think we're making a transformation at MIT as well. I sit in the School of Humanities, Arts, and Social Sciences. I was previously in the School of Architecture and Planning, and keep an appointment there. I think we're really figuring out the prominence and importance of the humanities, arts, and social sciences in solving the world's problems, and in concert with science and engineering. And so I look for more progress in that space. I think it's about about ways we come together to solve problems. It's about ways that we support students in doing that. We have extensive requirements for that now. Students take basically a course every year in that, every semester in that rather. It's about research support in these spaces, and so I think we can do more. But I think we've sort of we've recognized this, and I think it's a problem that we can work on together.
That's great. May, what do you think?
It's a hard question. I think MIT's best resource is the talented people, students, postdocs, faculty. So if the institute can find ways to better support students and faculty to do their best work, and maybe, I don't know, less administrative burden, or whichever things they can think of to support us, that would be really the thing that I would look for. So yeah.
Chris.
Yeah, I think if we could somehow embed the spirit of collaboration and can-do-ness back into the institute that we had during that little period. I mean, in our experience, we had, people said yes to foundation funding that required overhead that we might not normally be able to cover. We had nearly daily contact with research administration and the tech licensing office to make sure that we had all the paperwork to be able to ship strains all over the world and do things to share the research that we were doing. So there was continuity in all of those aspects and how we operated through the infrastructure that felt like a team, as opposed to I'm going to throw something out in the ether and see if maybe someday I get an answer on it.
Yeah.
So if we can find ways to kind of come back to the infrastructure side and kind of recapture aspects of that, and everybody wanted to do it, right? So I think some of that may have to do with even education of our own whole community of how we are transforming the world through the things that we're able to do together.
Yeah.
I think those would be things that might come to my mind.
Yeah, yeah. No, I agree. I think my experience was similar in these models that Ian talked about at the beginning. This was like you said, everybody wanted to contribute, and we sort of threw out the normal research model where I work in my lab, you work in your lab, you work in your lab. It's like, okay, this is all hands on deck. We all know the problem we have to solve. We have a limited time to solve it. Tell me what you got. And there was something about the immediacy of that crisis that sort of all of anything that you would normally get tripped up on was just gone. It's like we're all rowing in the same direction, and we know it has to be done. So I agree. I think it was a really unique experience, and I do think we need to think about ways that we can preserve that moving forward, because I think, honestly, I couldn't have been more proud to see the way the community stepped up, and I couldn't be more proud of what MIT did during the pandemic. So and with that, I'm 59 seconds over. So I would like to thank our panelists for a terrific conversation.